Shear stress effects on epididymal epithelial cell via primary cilia mechanosensory signaling. Sounds complex, right? But what is this paper really about? Imagine yourself on a beach. The sun is shining, the sand is soft, and you decide to take a swim. Wouldn't it be crazy if simply being exposed to the flow of water could teach you a new skill? Now, what if I told you that something similar might be happening in the male reproductive system around puberty? This paper, published in the Journal of Cellular Physiology, explores how shear stress, a force generated by fluid flow, could give new skills to the cells involved in the control of male fertility. You see, before boys hit puberty, their reproductive organs are waiting for a signal to start maturing. One of the main such signals is testosterone, the male hormone produced in the testicles. But there are plenty of other signals, and the physical effect of fluid flow might be one of them. How do we know? In this study, researchers examined the effects of fluid flow on the cells of the epididymis, a narrow coiled tube attached to each of the testicles. As fluid from the testicles begins to flow through these tubes, it can trigger important changes in gene expression, influencing the proteins that cells produce and, consequently, the functions of the cells, or their skills. But how can fluid flow outside the cell teach a cell new skills? Further analysis revealed that these cells have a tiny hair-like cellular antenna projecting into the lumen of the tube, sensing the fluid flow. This antenna, called the primary cilium, bends under the pressure of the fluid flow. The shear stress bending the primary cilium initiates a cascade of intracellular events that eventually reach the DNA, modulating the proteins that cells produce and the cell's functions. So, now you know what are the